Okay, so the data we're going to look at is actually data for my thesis, which is looking at the effect adolescent nicotine exposure has on dendritic morphology of the bed nucleus of the stria terminalis. So a little background is the bed nucleus plays a role in addiction and anxiety, and anxiety is seen normally in adults who smoke during adolescence, and clearly addiction is formed through adolescent smoking. So that's just your background. For this study, we had 16 subjects, and all the data was observed for all of them, so there was no problem of missing data. Had there been missing data, as stated in the article by Eva Petkova, missing data would then create barriers to inference about the magnitude of treatment effects in a randomized experiment. The main concern of missing data, as stated in this article, is that analysis of data with missing information, there's a possibility that the missing observation might differ in an important way from the complete observation or the observations that you do have. But since all of my data is present, our inferences should be fairly on point. So when we look more at my data, all the subjects are numbered and identified in the subject column. So we've got A1, A2, and then it goes all the way down to E4. Oops. So E4. Okay. And then each subject is identified as receiving either saline, which would be 0, or nicotine, which is a 1. Now we can see that there are multiple entries for each animal, so there's like 15 entries for animal labeled as A1. This is because 4 to 6 cells were reconstructed from the bed nucleus of the brain, and they're reconstructed for each animal, and data was collected about each cell according to 3 different parameters. So each cell collected has 3 entry points. So there's a first parameter, which was the number of branches as a function of the branch order. So we identified branch order here, and then this is just the number of branches that occurred at each branch order of cell 1. And then it goes down here for the same thing for this cell 2, and so forth. The second parameter was the total length of the dendrite as a function of the branch order. So for each branch order, they get a length. If they didn't have that branch order, then there's just a 0 entered. And then the third parameter is the average length as a function of the branch order. So if there are 3 branch orders 3, 1, that would be the average of that. So that's average length for each branch order. But for my thesis, a mean value of each of the morphological measurements of the 3 parameters was calculated for each animal. So statistical analysis was based on the mean value for each animal rather than on each cell being treated as an independent measure. So instead of treating each of these cells independently, we averaged all of their things and entered them into this as one cell, and labeled that cell as cell 999. So 999 is just the average of all the cells. And then since the averages, we want to make sure that when we're analyzing, we're getting the correct one. The parameters are then, the first parameter was the branching. That's now parameter 4. Parameter 2 is now labeled as parameter 5. Parameter 3 is now labeled as parameter 6. This is so when we want to select for only parameter 4, where we don't get individual cells as well. So the first parameter that we're going to look at is parameter 4, and we're only going to look at parameter 4 for the purpose of the module. So we went to data and did select cases and did if condition is satisfied. So if parameter equals 4, then we want to keep those cases. Otherwise, we don't want them. So that's why all of these are marked out, and we only have parameter 4. So when we're running our data, this is all we're looking at. Once we have parameter 4 measurement selected, I ran a repeated measures ANOVA on the data. And this is what I got. This first box is just showing that we looked at 5 different factors, and they were the 5 different branch orders. So branch order 1 through 5. This next one in between subject factors is showing us the differences between the two groups that we looked at. So the saline, which was labeled as a 0, there's 7, and then nicotine was labeled as 1, there was 9. Then we have our descriptives of the mean number of branches for each branch order according to the drug that they were administered. So for branch order 1, saline, there was a mean of about 2.9 branches that occurred in branch order 1. And then for nicotine, there was an average of 3.6 branches occurring in branch order 1. And then a total average of all the branches occurring was a 3.3. So this is just showing you just some more descriptives of what your data actually looks like. As noted in dealing with non-normal data article, you need to pay attention to your data's distribution, which can be done by looking at this box and seeing the standard deviations that are around the mean, and also by graphing your data to find out if they're normally distributed or not. Because if they're not normally distributed, then you have to take other routes in analyzing them. Alright, so the first thing we wanted to look at was our test of sphericity. So for this, if this is actually significant, then when we look down at our test of within subjects effects, we went ahead and corrected for it and used the Greenhouse-Geyser correction to find if there was a significant interaction. But for this parameter, we can see that the sphericity is not significant, so we can assume sphericity, wow. So we look at the branch average and the drug interaction, and we see our F value is 2.05, and then its significance value is 0.1. So it's not hitting the significant value that we want, which is 0.05. This could be due to the fact that my n is small, so there's only an n of 16, so the groups were fairly small. Maybe I had a larger experimental group or a larger just group in general, this significant value might be more significant, but for now, the interaction is not significant. So we then looked at the test of between subjects effects, and this is just looking to see if there was a significant overall effect of the drug on the number of branches. This is putting away branch order information. So we looked at drug, our F value is 23.315, and it is significant. So it's saying that when nicotine is introduced, there is a significant difference from 0 in the branching that occurs. So there's a significant difference in branching that occurs within nicotine as opposed to saline. So then we have our plot. This shows the differences between the branching of a saline subject versus the branching in a nicotine subject. I found this graph to be displayed fairly well and not poorly as described in the criteria of the How to Display Data Badly article, namely because, as I say, displaying data bad, you hide as much of your data that's shown. So this data is fairly well shown. There's no extra amount of information that's kind of fogging up what we want to look at. Our points are clearly indicated. There's a clear difference between the lines. We have a rule over here. It shows us that, yes, the blue line is saline, the green line is nicotine. And I also went through and relabeled them so they're even easier to understand when you're looking at it. So you know you're looking at differences in dendritic branching. You've got two groups that you're looking at, nicotine and saline. Over here is just the average number of branches. And then this is a branch order. So for branch order 1, an average of close to 3 for saline, and then close to like 3.7 or so for nicotine, and so forth. This also goes further with our idea that there was an overall effect, because you can see overall it is making a difference in all of these points, and it's not dependent on the branch order on making the effect. So even though there wasn't a significant interaction, we went ahead and ran a t-test to see if there were significant increasing of branches at a particular branch orders. So yet again, here's our group statistics. We saw these earlier, just saying the branch order 1, the mean of that for saline, and the mean for nicotine, and so forth. Now down here is our independent t-test. If the test of equality of variance is actually significant, then we want to correct for it by looking at the equal variances not assumed, and we would go across to there. So going through each of these for branch order 1, this equality of variance is not significant, so we're assuming equal variances. However, there was not a significant increase in branching in branch order 1. So branch order 2 we looked at. It is significant in the equality of variances, so we go down to equal variances are not assumed, and we see that there is a significant increase in branching in branch order 2. And then this isn't significant, so we can look at here, and there's a significant increase in branching within the branch order 3. And then this is significant, so equal variances are not assumed, and there is significant increase in branching in branch order 4. For this, there's a significant as well. Do equal variances not assumed. For branch order 5, it is approaching significance of increasing the amount of branches in branch order 5. These specific tables, I wouldn't exactly use them in my write-up, because according to the Rudiments and Numeracy article, a table is strong when its patterns and exceptions are obvious at a glance. And clearly, you would need a full background of the research that I did, and also a statistical background in order to even understand these tables. And for the purpose of my experiment, I find that it would be better represented in a graph such as this in order for you to understand my data. If I want to go back through and incorporate information that I found within my independent sample test and find that there's an increase in branching, significant increase in branching in branch orders 2 through 4, I can go through up here, and when I use this graph, I would just indicate by maybe an asterisk above each of these 2 through 4 that these increases and differences were significant. And that's all I have.